Have you ever thought about how a single cell, like smaller than the period at the end of a sentence, actually moves across your body?
It's pretty amazing, right? Like a microscopic explorer on a mission.
Totally.
Yeah.
It really makes you wonder what's going on inside to make that happen.
Well, a lot of it comes down to this protein called actin. It's like the cell's internal scaffolding.
Like the stuff they put up on buildings?
Exactly. But here's the thing. It's not static like regular scaffolding. It's constantly changing, assembling, and disassembling to move the cell forward.
So it's more like the cell is building its own little roadmap as it goes.
Yeah, that's a great way to think about it. Actin comes in two forms. You've got these individual units called G-actin, kind of like Lego bricks.
Oh, like that analogy.
And then you have these long chains called F-actin. Those make up the actual scaffolding. The cell can put these chains together really quickly through polymerization.
Polymerization.
Yeah, it's basically just a fancy word for building. And then it can also break them down through depolymerization.
So it's like constantly snapping those Lego bricks together and then pulling them apart again.
Exactly. And all that snapping and pulling creates forces that help the cell move.
Okay, so I get the building part.
Yeah.
But how does that actually make the cell move?
Well, think about it this way. If you want to move a piece of furniture across the room, you have to push it, right?
Yeah.
The same idea applies to cells. The assembly and disassembly of actin chains create these pushing and pulling forces on the cell membrane, which propel it forward.
Oh, so it's like the cell is using its internal scaffolding to kind of inch itself along.
Exactly. And what's really cool is that the rate at which those actin chains break down, what we call the depolymerization rate, actually has a huge impact on how fast the cell can move.
I would think faster breakdown would mean faster movement. Like get out of the way and go, go, go.
That's what you might think. But that's not always the case. We've been using mathematical models to study this. It's basically like creating a blueprint of how the cell works inside.
So what does your blueprint tell you?
It turns out that both very slow and very fast depolymerization actually make the cells slower. There's kind of a sweet spot in the middle where it moves most efficiently.
Really? So it's not just a faster is better situation?
Nope. It's all about finding the right balance. And this balance also plays a big role in how the actin is spread out inside the cell and how that affects its shape.
Okay, now I'm really interested. So how does this balance actually affect how the cell looks and moves?
Well, let's imagine the depolymerization rate is low. So the breakdown of those actin chains is pretty slow. Picture a really organized march, everyone moving in sync.
I can picture that.
In this scenario, the concentration of those F-actin chains, the scaffolding gradually decreases from the front of the cell to the back. So there's like a smooth gradient.
Okay.
And the actin is flowing mostly in the direction the cell is moving. So it's a nice, smooth, steady forward movement.
Gotcha. So it's like a well-oiled machine.
Pretty much. Now, what happens when you crank up the depolymerization rate? When that breakdown of actin chains gets really fast?
Does the cell become like a speed demon?
Not exactly. It's more like trying to walk forward while pushing sideways at the same time.
I could see how that wouldn't be very efficient.
Right. You'd spread out, but you wouldn't get very far. That's kind of what happens to the cell at high depolymerization rates.
So instead of focusing all its energy on moving forward, it starts to spread out.
Exactly. The actin chains get really concentrated at the leading edge of the cell, and then the flow of actin starts going more sideways, perpendicular to the cell membrane.
So it's like the cell is trying to spread its wings, but it's costing it speed.
Yeah, that's a great way to put it. So basically, the cell can actually change its shape depending on how fast it's breaking down those actin chains.
Wow. So it's not just about the rate of breakdown. It's also about how the actin is distributed within the cell.
Absolutely. And it gets even more interesting when you start to consider where the cell focuses its actin-building efforts. Not just how fast, but where, too.
Okay, my mind is officially blown. So if the cell can control both how fast and where it builds and breaks down this actin scaffolding, it's basically like a tiny little engineer sculpting its own shape and steering its own course.
You got it. And we can actually explore those sculpting and steering mechanisms in more detail using our mathematical model.
All right, I'm ready. Let's dive into this microscopic construction zone.
Okay, so we've talked about how fast the cell breaks down those actin chains, but what about the location? Like, where does it focus its actin building efforts?
Right, we were talking about how fast, but now we're switching to where. It's like you said, not just how fast, but where too.
Exactly. It's like shining a flashlight. You can make a narrow beam or spread the light out, right?
Yeah, like a spotlight versus a floodlight.
Perfect analogy. The cell can do the same thing with actin polymerization. It can concentrate its building efforts at one spot or spread them out.
Okay, I'm picturing it. So what happens if it focuses all that building in one tiny area?
Well, you get this concentrated force pushing the cell membrane outward. It creates a long, thin shape,
Huh?
like squeezing a toothpaste tube.
So the cell kind of stretches out.
Yes. Our model shows it actually forms something called a filipodium.
Filipodium. Is that like some kind of space alien?
Haha, not quite. It's basically a finger-like protrusion coming out from the cell's leading edge. They use it to explore their surroundings, sense things, and even pull themselves forward.
There's like the cell reaching out to touch and feel its way around.
Exactly. And that reaching is driven by that focused actin assembly we talked about, like a tiny little crane inside the cell extending its reach.
That's wild. Okay,
Exactly.
so focused assembly makes a long, thin shape. What happens if the cell spreads out its building efforts? More like that floodlight you mentioned.
Instead of a long protrusion, the leading edge broadens out like a fan. Imagine a car with wide tires. It's more stable but less agile.
So the cell is trading off agility for stability.
Exactly. And this wider shape often goes with a different type of movement called lamellipodial migration.
Okay, another new term. What's that?
Think of a slug slowly gliding along. It extends its front edge and pulls itself forward. It's like crawling.
So it's more of a gradual spreading movement, not like the focused reaching of the filipodium.
Exactly. And that difference all comes down to how the actin assembly is distributed.
This is blowing my mind.
Mm.
So the cell has these two different ways of moving, and it can choose between them depending on what it needs to do.
Exactly. It's like a toolkit. And it picks the best tool for the job. And here's the really cool part. Our model actually predicts these different movement patterns, and we've seen them happening in real cells too.
Wow. So it's not just theory. Your model can tell you what a real cell is going to do.
That's right. It's all about math. We build these equations that represent everything going on inside the cell, all those forces and factors like polymerization, depolymerization, actin concentration, the membrane, you name it.
Sounds complicated.
It is, but it's really powerful. Then we run simulations with different combinations of these things to see how they affect the cell's behavior.
So you're basically creating a virtual cell in the computer.
You got it. And the amazing thing is these virtual cells often act just like real ones.
That's incredible. So you can see how the cell will move and change shape before you even look at it under a microscope.
Exactly. And that opens up all sorts of possibilities. For example, we can use it to see how cells might react to different drugs or changes in their environment.
Wait, so you can test out treatments on these virtual cells before you even go into a lab?
That's the idea, like a dress rehearsal before the big show.
That's amazing. It's like you're peeking inside the cell and you can experiment with it in ways you never could before.
And those experiments can lead to some pretty big discoveries. For example, our model has helped us understand why some cancer cells are so good at spreading throughout the body.
Oh, wow. That's serious stuff.
Yeah.
Tell me more about how this connects to cancer. So we were talking about how your model can help us understand cancer.
Yeah, it's kind of a scary thought, right? How these same mechanisms that help our bodies heal can also be used by diseases.
Totally. So how exactly does cell migration, all this movement we've been discussing, connect to cancer?
Well, one of the big things about cancer is its ability to spread what we call metastasis. Yeah, basically the cancer cells break away from the original tumor and move to other parts of the body.
So it's like they're escaping from prison and setting up shop somewhere else.
Exactly. And unfortunately, these escaped cells are really good at migrating. They can squeeze through tiny spaces, navigate different tissues, even hitch a ride in the bloodstream.
So the same things that make them good at healing wounds and fighting infections also make them dangerous when they're cancerous.
Exactly. And that's where our model comes in. It can help us understand and maybe even fight cancer metastasis.
So I'm really curious to see how all this ties together.
Well, we can use it to simulate different situations and see how changes in actin dynamics, like those shifts in polymerization and depolymerization we talked about, affect the cell's ability to invade tissues.
So you can actually watch these cancer cells breaking free and spreading.
Pretty much. For example, we've seen that cancer cells often have more of certain proteins that boost actin polymerization. That helps them generate those pushing forces they need to squeeze through those tight spaces.
So it's like they're building their escape tunnels even faster and stronger.
Yeah, that's a great way to think about it. And if we can understand these mechanisms, then we can start to develop new treatments that target them.
So if you can figure out how they're breaking free, you can figure out how to stop them.
Exactly. Imagine drugs that can mess with those actin-promoting proteins, or maybe even tip the balance towards depolymerization. So it's harder for them to generate those forces.
Like cutting off their fuel supply or sabotaging their escape vehicles.
Exactly. And the great thing about our model is we can test all these ideas virtually before we have to do expensive and time-consuming experiments in the lab.
This is incredible. It's like you're leading a revolution in cell biology, using math and computers to unlock these secrets and find new ways to fight disease.
It's definitely an exciting time to be working in this field, and the more we learn about these basic processes, the more we realize what we can do with that knowledge.
Well, I'm so glad you're doing this work. It's fascinating and really encouraging to know that we're getting a better understanding of how cells work and finding ways to use that knowledge for good.
It's really rewarding work. And you never know, the next big discovery could come from anyone, anywhere. So keep that curiosity alive and never stop exploring.
That's a great message to end on. Thanks so much for sharing your expertise and giving us this amazing look into the world of cells.
It's been my pleasure.
That's all for today's deep dive. Until next time, keep those cells moving and stay curious.